So we are going to do into this particular video 6.3 and 6.4. 6.3 is RNA World. When the formation of Earth took place, and when very first living organism arose onto the planet Earth, which had the genetic material as the DNA. So there was a time in between, and this particular time, and sometime somewhere around here, there was a time period which is also known as the RNA World. Now the question is that why do we call it RNA World? Because onto this particular time period, we did not have, number one, there was no protein, there was no DNA, there was majorly nothing else was existing, and obviously there was no life as well. But there was a macromolecule, and this macromolecule was RNA. In today's world, the DNA and protein have got their specific work assigned to them. And what do? The protein actually is able to, the major portion of the protein is acting as the enzyme and the structure. So it is able to help you out, number one, to compose a structure, and number two, to catalyze reactions. Catalyze reactions. So these two are major work of a protein. DNA is acting as the storage information. Okay, so it is the information which is able to, which some of the protein are able to read from the DNA, and they are able to make some other catalytic protein, some other different kinds of proteins. So no DNA. So the DNA was carrying out the information. So information. Now all these three properties were carried out by RNA. So this is why we call that period as the RNA world. Now in today's time point, do we have any major evidences that by which we can say indeed RNA can act as, like, it can fulfill all the duties of a structure, catalysis and the information? Indeed. And that particular moiety that is indeed that exists in today's time point is known as the ribozyme. Okay, so as you can see that the name itself is saying too much, that it is a ribozyme. So ribozyme is RNA, and this particular RNA is able to take out a specific structure, and this particular structure is able to do all three duties. And after some time, this RNA actually evolved into DNA, and DNA was made an integral part of a cell, and there comes the very first cell with its double-stranded DNA, and the role of RNA is now was no more. So it is able to tell you that RNA was the first genetic material. We just get to know that why and how it can be the very first genetic material, as it was able to fulfill all the duties of structure, catalysis and the information. So therefore DNA has evolved from RNA with chemical modification that make it more stable than DNA. Okay. Now let us start 6.4. One of the major portions of this particular book are three things: replication, transcription and translation. How you are able to take the information from the DNA, and if you are able to convert it into a form of product, and that product will be the protein. We know cell divide all the time. One cell can become two cell, and this particular process is known as cell division. But the interesting point to notice is that the parental cell also have 23 pair of chromosome, and the daughter also have 23 pair of chromosome. So there must be a process which is lying in between, in which this genetic material, which is 23 pair of chromosome, is getting duplicated, and this very process is known as replication. But before going into the mechanism of the DNA replication, let us go through one particular experiment. And this experiment is about that what is the mode of this particular kind of replication. So if this is the parent DNA, now the question in front of the scientist was that how is it replicating? There can be three methods for that. Either the parent is remaining intact and the new DNA which is getting synthesized is totally made up of a new monomers of DNA. Then this is known as the conservative mode of the replication. Or what second thing can happen, that one parental strand is remaining into the progeny, and onto that particular strand a new strand is getting synthesized. And this kind of replication mode is known as semi-conservative mode. Semi-conservative. And as you can see that the parental strain is remaining conserved into the conservative mode, and the same into the semi-conservative, half of the parental strand, half of the parental DNA is getting into the progeny. Now what could be the next type of mode of replication that before the experiment scientists hypothesized? That it could be the mixed one, in which the parental as well as the daughter DNA is getting somehow mixed up, and you're getting a mosaic kind of a pattern into the DNA replication. Now let us go to the experiment. The Michelson and Stahl grew some bacteria onto a Petri plate. But onto this particular Petri plate, they had a normal food. Let us say this was the normal food. And this normal food had the normal nitrogen. But they wanted to incorporate, they wanted to change all the normal nitrogen 14 which is present into the DNA. They wanted to change it with the nitrogen 15, with a radioactive nitrogen. For that, they took the bacteria from a normal plate and they basically grew it, this very tiny fraction of the bacteria, they grew it onto a new plate. This new plate had radioactive nitrogen as the nitrogen source onto the food. So consecutively, what happened? Now all those bacteria which were living onto this particular plate, they incorporated nitrogen 15 into their DNA. Now they took a little bit of the bacteria from the nitrogen 15 plate and they plated on some other plate. This plate had the food with nitrogen 14 intake. They transferred some bacteria from the plate which was radioactive to a new plate which carried normal nitrogen. So this was our 0.0 time, and this is also our 0 time. Now they waited for 20 minutes right here, because one replication or one cellular division into the bacteria, into the E. coli actually takes 20 minutes. Then it makes sense that they allowed these cells to divide only once. After this, they also waited for 20 minutes more. Since the scientists could not see all this process going on directly under the microscope, they were relying themselves onto some molecular techniques. Now this is the technique. They took the samples from each of the Petri plate. Now let us see. A little bit of sample was taken from this particular. Since they could not see this particular process under the microscope directly, they were relying themselves onto some molecular techniques. These molecular techniques were that they took sample from each of the Petri plate and they applied and they subject all these test tubes to a centrifuge. After taking samples from all the plates, they added the lysis buffer. And when they added the lysis buffer, the cell burst open. And all the DNA material that was there inside the cell, now it was inside the solution. Okay, so let us try to analyze this result. Number one, we have got a Petri plate, and into this particular Petri plate we have got our nitrogen 14 environment. So all the DNA or the genetic material that will have nitrogen 14 incorporated in it. Now what we are going to do, we have taken a test tube, and apart from this test tube we took some of the colonies from the petri plate, we added the lysis buffer, and after adding the lysis buffer all the DNA is going to spill into the solution, and we are going to subject this test tube to the centrifugation. And after the centrifugation, what we are going to get, we are going to get a bend, and this bend will, let us say that this band, and we are going to subject this test tube to the centrifugation, and we are going to get a particular band, and this band is going to settle itself according to the gradient of cesium chloride. Okay, so we have got a cesium chloride gradient into this particular direction. So according to the cesium chloride gradation, it is going to set itself. Let us say since it is lighter, so let us say that it has settled itself somewhere around here. Now what is going to happen into, let's say, test tube number B, that since all the DNA is radioactive and nitrogen 15 is heavier than the nitrogen 14, we are going to get one band, but this one band is going to be present somewhere around here because it is heavier. And this is the important point. So I am going to get a mixed band which is going to lie in between, okay, which is going to lie in between your red one and the green one, because they both are composed of nitrogen 15 as well as nitrogen 14. And as far as this third test tube is concerned, that what is going to happen, that you are going to get one particular band, very nice prominent band of nitrogen 14, and again one mixed band, because this mixed DNA, sorry, no, it is not, but semi-conservative DNA is present right here. Now next question is, I gave you some other replication methods as well. What if they turned out to be true one? So for them, if they were the true, what would be the result outcome had been? So this is what it would have looked like. Into the test tube number 1, again we'll have a normal same nitrogen 14. I'll have nitrogen 15 as well. So let us say this is the test tube number 2, 1, and for the 2 we'll have again the same gradient. But for the third one, since this is conservative, again we will get two distinct band, one of the nitrogen 14 one and one of the nitrogen 15 one will not get an intermediate band as it happened into the semi-conservative mode. And that is why we said that indeed this is the method by which DNA is able to replicate itself. It is able to replicate itself by the semi-conservative mode. The same thing is going to remain into this band as well, that we'll have a red one right here, it's a heavier one, and a green band right here. Now let us say that if it was mixed, okay, so let me remove it. Okay, if it was mixed, then this would have been the condition. For the green one, no problem, we'll have a lighter band. For the nitrogen 15 as well, we don't have any problem, we'll have a, and after that, since it was mixed, we'll always get this one, the blue one, the mixed one, like this. Okay, so this was the semi-conservative mode of replication of DNA by experiment. Let us move on to the mechanism itself. Now let us start with the replication of the DNA, the mechanism of the DNA replication itself. If this is my DNA, then the very first thing that I need is to open it up. And to open up a DNA double helical structure of the DNA, we have got an enzyme which is known as helicase. This structure is known as a replication bubble. The next protein that comes, if we try to zoom in this particular place, then we are going to see that the very next enzyme that comes is known as primase. And primase adds a little bit sequence of oligonucleotides that is made up of RNA onto the DNA primer, and this particular sequence is known as primer. After this primer comes our main DNA polymerase, which can polymerize the DNA. So here is our primer, and primer can be polymerized by DNA polymerase. The drawback of the DNA polymerase is that it is not able to start the synthesis of the DNA itself. So that is why it needs the assistance of the primase, so that primase can go and attach a bit of oligonucleotide over there, and that oligonucleotide, since it has got that free 3-OH group, which it can polymerize very easily. But just as I mentioned that it is able to take the existing OH group and it is able to add a new nucleotide onto this OH, 3-OH. So this is its specificity. It is not able to, that is why we always say that it is able to make the DNA strand from 5' to 3', but it is unable to do so into the opposite direction. So it is unable to make the DNA into the opposite direction from 3' to 5'. So just as we have added primer, if we can say that this was our 3' side, this was 5, this is 5', this is 3', then DNA is able to polymerize from 5' to 3' direction, but it is unable to do so into the opposite direction. That is why we will not able to get a DNA into this particular direction. So what is going to happen onto the opposite strand, because as we know that we also need to make the opposite strand also equally good. So for that thing, that the primer is being added onto several different times. So onto this particular strand, which is onto which the synthesis of DNA is taking place flawlessly, and that particular strand is known as the leading strand. And onto the opposite strand, the primer is being added after several places like this. So the DNA polymerase can come and it can polymerize these strands as well. And this is known as the lagging strand, because as you can see that the continuous DNA replication is not taking place. And when this polymerase is able to polymerize this particular primer, this polymerase also cleaves this primer which is present already, and it replaces it with the normal DNA. But still, in spite of this fact, there is a gap in between, and this gap is being sealed by another enzyme which is known as ligase. And DNA replication cannot start by itself at any place. Why? Because the assembly of different polymerase needs to happen, and let us say that this is a very specific nucleotide sequence which is able to recruit DNA polymerase, primase and other enzymes. So this is known as the origin of replication. Origin of replication, or into the short form it is also known as ORI. E. coli, since it has got the circular chromosome, that it has got only one ORI, but since we have got linear chromosomes, we have several different ORIs. So from the level of NCERT we are done. So as you can see that as far as these terms, that we are done with the replication fork. So this bubble, if you try to see this bubble onto this half side, will look like as a fork. So this is known as the replication fork. The continuous strand is the 5' to 3' strand, which is moving from 5' to 3' direction, and this continuous strand is the lagging strand. Okay, and thus the gap that remains between the previous coming DNA polymerase and it is coming towards the primer which was added before, this particular gap is being sealed by the DNA ligase. And the origin of replication, as we have just discussed, that the sequence onto which the polymerase assembly and the protein assembly actually is being recruited, and from there the replication of DNA started. That place, that origin of replication, as the name itself is self-explanatory, is known as the ori.